It's really an amazing little critter, and it's been around for over 3 billion years. In 2016, Utah Lake exploded in cyanobacteria blooms. The problem is that many cyanobacteria produce toxins.
You may have heard it called blue-green algae, but it's really a kind of bacteria, taking in sunlight to drive photosynthesis and giving off oxygen.
It actually requires quite a bit of lab testing to know whether or not it's a harmful algal bloom. And what we're really worried about is people and pets ingesting that cyanobacteria.
Dr. Kate Fickus is a harmful algal bloom scientist at Utah State University. She helps the Utah Department of Environmental Quality track conditions in lakes and reservoirs.
So Utah is the second driest state in the nation. Most of our major lakes are actually man-made reservoirs. They're heavily used for recreation, they're heavily used for agriculture, and they're really important to the state as a resource.
In 2017, harmful algal blooms returned to Utah Lake. This time, officials used satellite data to identify troubled locations. But how can instruments up in space tell us about microscopic organisms in a lake down on Earth? By measuring their blue-green color. Landsat collects light in visible and infrared wavelengths. Cyanobacteria reflect more green light than plain water does, allowing Landsat to identify algal blooms.
From satellite, what we see is basically that primary pigment, which is chlorophyll A, but the color by itself could be misleading. A nice picture is not necessarily providing a set of quantitative data.
Algal blooms can look beautiful from space, but the numbers behind the images are the important part.
Each measurement is highly accurate, and it's very much corresponding to the number of photons that are leaving the body of water, which could be related to the biomass and the amount of phytoplankton.
Dr. Nima Pahlavan is working with NASA and the U.S. Geological Survey to make sure Landsat users have consistent, accurate, and ready-to-use data about lakes and rivers. Water is difficult to study from space because only a fraction of the sunlight is reflected back to the satellite. But engineering improvements on Landsat 8 have leveled up its ability to measure the small signals from water bodies. After Landsat collects the data, it gets beamed down to the USGS AROS Center, where it is archived. The raw numbers pass through checkpoints to align the geography, correct for sun strength, and then compensate for the effects of the atmosphere.
So you essentially removing those atmospheric scattering and absorption.
Let's break down what NEMA means here. To measure the amount of cyanobacteria, you need to know how much light reflected off the surface. But some of that light gets scattered by molecules in the atmosphere on the way to the satellite, lessening the signal received. And sometimes light that never made it to the surface gets scattered into the satellite, adding a false signal. Like removing the haze from a photograph. Atmospheric corrections leave you with a quantitative measurement of exactly how much light left the water, known as aquatic reflectance.
You want to look at the actual physical measurements to derive physically meaningful products from satellite data.
And that's the goal, to transform the raw materials into finished products so that end users don't have to build it themselves.
By providing aquatic reflectance products, you're majorly reducing the burden on satellite users.
Although it is still provisional, NEMA's aquatic reflectance data product is available for download from the USGS. Scientists like Kate Fickus convert the data product to maps showing the amount of chlorophyll A, helping local officials pinpoint where to test for toxins and warn residents.
I use Landsat and other remote sensing technology to help local health departments understand where there's a bloom, the magnitude of a bloom, and the size of the bloom.
The spatial detail is another benefit of using Landsat data. Each pixel is only a 30-meter square, the size of a baseball diamond, yet it collects data across a broad area. In other words, there is a lot of data at a fairly high resolution.
With Landsat, we can get into some of the marinas that are popular fishing and swimming spots in order to inform local health departments about making public health decisions.
For the 2017 outbreak, that's exactly what happened. Satellite data gave an early warning to local officials in Utah. The extra week of warning saved hundreds of thousands of dollars in healthcare costs. Monitoring algal blooms from aquatic reflectance data is just one example of benefits from Landsat's data products. Wildfires, snow cover, vegetation health, temperature, and more are available for every spot on Earth. Landsat's highly calibrated data products, free to download and use, are making detailed Earth observation data more accessible to users and bringing a greater benefit to society.
Well, hello everybody. Happy Friday. Today's a kind of a crazy day here. We start off with the beautiful weather, and then suddenly we have this huge downpour of rain and stuff like that, which is, for if you're going to go hunt for microscopic animals and stuff like that, specimen hunting. It's best right after a rain because a lot of stuff comes out. Now, I was busy today preparing for the Astronomical League's live number 6 program, which is going to happen here tonight. But I was able to put together some stuff that I had been working on, which was kind of learning more about the history of the development of the microscope, which we covered in last week's program, and now I'm kind of diving into the individuals and the development of learning about biology, and it's very interesting that what people thought. How people thought life evolved, what people thought of, for example, like fossils, the kind of the big gaps also of making some discoveries, yet being able to act on those discoveries for the benefit of humans. So that's very interesting to me as well. I'm learning a lot and I'm having a blast. But I did want to recognize the people that are on with us today. We have James, the astrophotographer, saying Happy Friday, and Cameron Gillis, Happy Friday as well. Mike Wiesner, just another Friday. Oh, okay. So we've got Harold Locke, Happy Microscopy Friday. Yes, that's right. And Cameron Gillis, Jeff Wise is with us, and I can't read this, but it says, "Hey, I live like a mile away from Utah Lake." So, very interesting program about the algae and stuff like that, and the blooms that happen. To me, a lot of this stuff that they study at NASA. Certainly lends itself towards the study of astrobiology, and so there's a lot of cross interests and the need to share knowledge between the biologist and the planetary scientist, for example, in order to try to find the search for life on another planet. Harold Locke says, I was replying to Mike Wiesner. I was already for cell phone astrophotography and got totally sucked in. I'm sorry, dude. And James, the astrophotographer, says cyanobacteria can kill off a coral reef ecosystem incredibly quick. Yeah, it's it's a dog eat dog world, or a microbe eat microbe world out there in the world of microscopy. And who else is with us today? Of course, Jeff Wise. Pekka's on with us today. Hi, Pekka. How are you? And CFox0011 says every Friday is a gift. I would agree with that. So before I dive into my presentations and stuff like that, I do want to, show the equipment that I'm using. This is a Bresser Science Infinity microscope. It's a biological microscope. I have a dark field adapter that I'm learning how to use, but you can see there's a camera up on top, a binocular head, and this is called a trinocular head, actually, and it works out really well. I mean, it's for a beginner like me, it's a solid instrument, good optics, and very versatile with like LED illumination and very precision mechanical stage and all of that. So. And I'll show you an image, a video capture I did of something called a copepod, which will come up here in, well, I guess it comes up right now. So we'll show you the video that I did, and then I'm going to jump to my presentation about the world of microscopy here. So, but this is the video that I did. Now, this is a crustacean, and right here at the head, you can see there's like a little black spot. Depending on the illumination that I used and the contrast that I set and saturation and stuff, this actually appeared red, and it is an eye of sorts. One of the things that I learned about copepods is that most of them, I think there's only one species out of all of them, and there's a lot, they don't even have a heart. There's no vascular system in a copepod. So, I'm not saying that they're a cold-blooded sort, but, they're a no-blooded sort, so how's that? So, but they were really interesting, and they sit there very, very quietly, and then they'll move. But in the video that I show here, you can see some movement inside here, and I expect that is digestion going on within this copepod. And what did we learn here, or what did I learn? It's not what you learned, but let me share my screen here a little bit. I really need two screens to do this. But let's see. Okay. I'll take that up full screen. Change what I'm sharing. Here we go. Okay. All right. So just like I feel like I'm in high school or something, or maybe even junior high biology class. But copepods are crustaceans, and there's freshwater varieties and saltwater varieties as well. And they have the potential to act as a biological control for malaria because they eat mosquitoes. However, they are also an intermediate host of many other animal parasites that might be harmful to us, including, they said, the fish tapeworm and the guinea worm. That does not sound good, but anyways, I'm careful when I put the copepod onto the microscopic slide. They say they vary considerably, but can typically 1 to 2 millimeter. The ones that I was observing were not even that big. And they have a teardrop-shaped body. They say and they go on to say that they have large antennae. but look at these things. This is some different types here. The one that I have is called a cyclopodia. Okay, that's what I took images of. But I mean, these things look really strange and unusual, almost like aliens from another planet. I will get to my the rest of my presentation here. I want to talk. I started getting, learning about the optics of microscopes, and of course, I understand the mechanics of microscopes, but a lot more fascinating is what people were learning, and so. And before microscopes were really invented, there were ideas that invisible organisms, things that we just could not see, were causing disease. And this is attributed to, this idea is attributed to an astronomer, and a physician, Girolamo Francastro in 1546. So he wrote this book about it, on contagion and contagious diseases, and stated that diseases were caused by a different type of rapidly multiplying minute body. He's describing bacteria, I think, and that these bodies are transferred from the infector to the infected in three ways: by direct contact, by carriers such as soiled clothing and linen, and through the air. What a revolutionary idea, okay? And why am I not getting to my next screen? Here we go. So we talked a little bit about Antony von Leeuwenhoek being credited with developing one of the first high-power simple microscopes. This guy, because he was able to get magnifications up over 200 magnification, actually let him see or to see bacteria on cloth. He was a drapery merchant, as I mentioned last week, and he wanted to get the finest threads for his drapery, and he finds just bacteria. So this goes back to our friend Castro saying that this stuff could be on clothing and linen and through the air. So this is something to keep in mind if you're trying to keep away from any kind of infectious diseases. You want to wear clean clothes, okay? and you'd like to wipe down surfaces as much as you can. Keep a clean house, it's going to make you less sick. So anyhow, Leeuwenhoek, he's not the first guy, but between him and another guy named Robert Hooke, these guys are the first to see microorganisms and to, among the first guys. I imagine there's others that might have observed them or, had some sort of experience to detect them. But, the deal is that these two guys wrote it down, published results, and get a lot of the credit for developing the entire field of biology and microscopy. So, an exciting times. But these guys are fighting against thousands of years of ideas like spontaneous generation. Now spontaneous generation is not a thousands of years old hypothesis, but the idea the example here is that spontaneous generation was that, if you took pieces of cheese and bread and wrapped them in rags and left them in a dark corner, that, you'd come back later and find that there were mice in the rags. Well, ta-da, that must have spontaneously occurred because of cheese and bread wrapped in rags, So, very interesting and funny in some ways to me and to others, when you think about that's how people thought, Leeuwenhoek called his microbes dirtjes, I think is how it's pronounced. That's Dutch for small animals. In English, they're called animalcules, animalcules. And of course, we would just call them, biological, microscopic biological microanimals, I guess would be the most contemporary description. Robert Hooke, this is an illustration of him. This guy's really amazing. He's an astronomer. I mean, all the science this dude did was just incredible. He makes, I think, distance measurements to I think he's the first one to make a distance measurement to a star other than the sun. Okay, so, they definitely an amazing mathematician and scientist overall. And he wrote a book that encompassed many of his ideas. It was called Micrographia, cool name for a book. But he incorporates his discoveries in the microscopic world and the astronomical world as well. He is the first to coin the term cell, suggesting that the plant structures resemblance to honeycomb cells. And he is also, I found this interesting, his observation of microscopic structures in fossilized wood led him to conclude that petrified wood and fossil shells were the remains of living things that had been soaked in he describes petrifying water laden with minerals. Okay, voila, we get fossils. But he, of course, he opens up the whole field of the past history of life on Earth, and very, very interesting. This illustration that you see of the microscope here is actually still on display. They still have it, and I've seen photographs of it. It's quite beautiful, and so I always love those antique scientific instruments. And so this is the gap that I was talking about earlier. We go from the 1600s, and now it's well into the 1800s that that theories of like all organisms are made of cells, and the idea that of germ theory of disease would prevail, versus and totally destroying the idea that spontaneous generation or spontaneous life erupting from non-living matter could occur. And so Louis Pasteur put the nail in the coffin on that particular idea by making an experiment where he boiled water and sealed it up, and was unable to get any kind of spontaneous life to emerge from it. Let's see, let me take you back. Let's find my video, please. Here we go. I'm back, folks. Anyways, it's this learning process of how to, photograph through a microscope and trying to get the best kind of images, and then kind of trying to take a dive back into history of what led up to modern biology today, and the discovery of life and past life and all of this stuff, and how astronomers played a huge role in all of this stuff is pretty amazing to me. Leeuwenhoek himself was not an astronomer. He was more of a merchant, but made a huge contribution to science. But many other astronomers were very much into microscopy, including guys like Galileo and many others, all the way up to George Ellery Hale, whose most treasured possession, even after building 4 of the world's largest telescopes in succession and all the contributions that he made to to the field of spectroscopy and the development of astrophysics and everything. His most treasured device was this microscope that his dad had given him. So I think that I'm kind of I'm trying to relive some of that myself, and it's a kind of a fantastic voyage. So, and which is also one of my old favorite movies. I think it was called Fantastic Voyage, where people were miniaturized and injected into somebody's body. So as they flew around in a tiny spacecraft, kind of predating the idea of injecting nano machines into someone's body to kill off disease and stuff like that. So anyhow, it's a pleasure as always to see you guys, and thank you for watching, and thanks for going on my my personal journey through all this stuff. And I hope to make some better images. The next video I'm going to try to do a dark field again with the Copepod. I find those things very fascinating, and we will see you later tonight. We have at 6 p.m. I believe. It's 6 p.m. Central is the Astronomical League Live number 6, and that's so I will see you at that time, and until that moment, you guys keep looking up, and thank you very much for watching. We'll talk to you later. Hey everybody, Scott Roberts here with Mike Hatch, and we're here at Explore Scientific Live, and we wanted to show you the Bresser line of microscopes. Now we're just showing a few of them here. We've got microscopes that do fluorescence work, some used for metallurgy. A microscope like this can look at 3D objects, including things like circuit boards, or if you have insects or anything like that, this does an amazing job. A lot of these microscopes can also take a camera attachment, which is great.
Like these right here with the trinocular.
That's right, and we have a special camera that does that. Some of these other microscopes have a way to attach like your iPhone to them so that you can get images. This one right here has a built-in monitor, so it's pretty cool. The microscopic world is endlessly fascinating, just like astronomy is, and we hope that you take a look.